अब हम बात करेंगे रीएजेंट्स और सप्लाईज की जो हमें चाहिए टू परफॉर्म एन एलाइज़ा। 96 वेल प्लेट पॉलीस्टाइरीन की ज़रूरत पेश आएगी टू परफॉर्म द मेथड। उसके अलावा हमें डिफरेंट बफर्स चाहिए टू परफॉर्म द स्टेप्स, like if we have to coat the wells with the antigen or antibody, we need a coating buffer. Coating buffer is phosphate buffer that contains the sodium phosphate and sodium chloride and is prepared at neutral pH, that is almost 7.2. This coating buffer can be prepared as per given recipe, and the volume which is initially prepared is 1 liter, which can be stored at 4 degrees centigrade and it is stable for 2 months. अगर volume required देखी जाए for 1 plate of 96 well, we need around 12 milliliter of this buffer to perform the coating step. Then we need washing or dilution buffer. Sometimes we need to dilute the samples. For that purpose we are going to use the same buffer, and we have to wash after every step during the procedure. For that purpose we are going to use the same buffer. हम इन buffers को classify करते हैं buffer A, B, C and D के reference से. So this is referred as B buffer, which is again a phosphate saline buffer that contains the sodium phosphate, two types of sodium phosphate, then sodium chloride, but it also have tween 20. Its composition is slightly different from buffer A. It contains tween 20, which is not part of buffer A, and then the quantities which are going to be used or the molarities, they are different. Again the same thing, this buffer can be stored at 4 degrees centigrade and is stable for 2 months. Then the third one is the chromogenic buffer or chromogenic substrate buffer, buffer C. This buffer is going to be used to develop the color, which is the basis of detection. For that purpose there are tablets available or there are ready to use chromogenic kits available, which can be used for preparation of this buffer, and this buffer also needs the hydrogen peroxide to be part of this detection or chromogenic buffer. इस बफर या इन बाकी बफर्स के हवाले से अगर देखा जाए, जो A और B बफर्स हैं, they are stable and they can be prepared beforehand, but buffer C needs to be prepared when it needs to be used. Always freshly prepared it. और इसकी प्रिपरेशन के लिए जो भी ग्लासवेयर हमने यूज़ करना है, अगर बेकर है या फिर किसी और किस्म का ग्लासवेयर यूज़ कर रहे हैं, so it should be washed, that the glassware should be clean enough, and any kind of contamination can be visualized when we prepare this buffer and we add hydrogen peroxide. Then we watch its color. Its color should remain slightly yellow. If the color changes from slight yellow towards dark yellow or variable intensities of yellow, that indicates the glassware was contaminated and this buffer can no more be used for ELISA test. When we add the hydrogen peroxide, we have to wait for some time so that the temperature of buffer comes to the room temperature. Then we need to prepare 0.5 molar sulfuric acid. Sulfuric acid and water is going to be used for this preparation. इसमें एक चीज़ का ख्याल रखना है कि हमें वाटर को एसिड में मिक्स नहीं करना, बल्कि we have to mix 28 milliliter of sulfuric acid in about 900 milliliter of distilled water. So water is going to get the acid. We are going to pour acid into water instead of we are going to pour water into acid, because if we pour water into acid, it can cause boiling and it can be problematic for the or dangerous for the researcher. Later on, we adjust the volume up to 1 liter with distilled water. The other components which are required, the antibody, enzyme, substrate, and the test sample.